Good morning, student. Today we are going to know about chloroplast structure or a chloroplast which is used as a photosynthetic apparatus in a photosynthesis when there is a development of organic food material. So first of all, chloroplast is a cell organelle which is present in plant cell. That is the most important difference in between plant cell and animal cell. In eukaryotic organism, the photosynthetic organelle is chloroplast. In case of dicotyledon, chloroplast can be seen with a light microscope and appear as biconvex disc about 3 to 10 micrometer in diameter. There may be only a few chloroplasts in a cell or as many as in some palisade, as many as 100 in some palisade mesophyll cells. So here is the diagram. That is the transmission electron micrograph of chloroplast. Sometimes the chloroplast is present in not just only in a leaf cells, but it also present in a stem which are having green in color. So the structure of chloroplast is shown just now. Then each chloroplast is surrounded by an envelope of two phospholipid membranes. A system of membrane also runs through the ground substance or stroma. Means inside chloroplast there is a substance which are present at inner wall called as a stroma. The membrane system is the site of light dependent reaction of photosynthesis. It consists of series of flattened fluid filled sacs called as a thylakoid. This is the most important in case of photosynthesis. So this thylakoid is placed in the form of stacks called as a grana that are joined to one another by membrane. This membrane of the grana provide a large surface area which holds the pigment, enzymes and electron carriers which are needed for the light dependent reaction. The membrane make it possible for a large number of pigment molecules to be arranged so that they can absorb as much light as necessary for the photosynthesis. Then the pigment molecules are also arranged in particular light harvesting cluster for efficient light absorption. So first of all, pigment molecules are also arranged in particular light harvesting cluster of efficient light absorption. In that sense, these pigment molecules are most important for the trapping of a light. So when they can trap, they can hold or absorb whatever light it gets. After that, that light energy is converted into a organic food material, and for that purpose in each photosystem the different pigments are arranged in thylakoid in funnel like structure. So here is the diagram. that is 13.2. So we are going to see that diagram. So here is this. So this is the diagram which shows a diagram of chloroplast. Diagram A is nothing but a chloroplast, while the diagram B is a photosystem, a light harvesting cluster of photosynthetic pigments in the chloroplast thylakoid membrane, and only a few of the pigment molecules are shown over here. So this is the light which are going to be trapped over here. This is one another light. This is the complete reaction done in a stroma, and this is the thylakoid membrane. So inside the thylakoid there is a presence of stroma, and within that there is a trapping of two different light. So this is the complete structure of a chloroplast. Each chloroplast is made up of by the outer and inner membrane, that is called as a chloroplast envelope. Then chloroplast also possesses its own DNA, so that is a chloroplastic DNA. And then there is a ribosome, starch grains, granum, and these are the thylakoid, and whatever the long elongated filaments which are connect two different thylakoid, that is a lamella, and whatever remaining fluid is called as stroma. So what happens when there is a chloroplast which is participate in a photosynthesis for the development of a food material? At that time, each pigment passes energy to the next member of the cluster, finally feeding it to the chlorophyll A reaction center, and that is called as a primary pigment. Later on, the membranes of the grana holds ATP synthase and are the site of ATP synthesis by chemiosmosis. The stroma is the site of the light independent reaction. It contains enzymes of the Calvin cycle sugar organic acid. It bathes the membranes of grana and so can receive the products of the light dependent reaction. So this reaction is completely light dependent reaction. Also within the stroma are 70s ribosomes, a loop of DNA, lipid droplets, and starch grain. The loop of DNA codes for some of the chloroplastic proteins. So first of all, this chloroplast possesses its own DNA, that is chloroplastic DNA, which can develop or which can produce its own proteins for the energy requirement, which are made by the chloroplast ribosome. However, other chloroplast proteins are coded by the DNA in the plant cell nucleus. So, in this way, this chloroplast is participating in a photosynthesis with many more functions. Then these are the factors necessary for photosynthesis.